All right, let's dive right into this explainer. We're looking at something literally happening inside you right now. How a single cell perfectly copies its DNA to become two completely identical cells. It's essentially a beautifully choreographed biological dance. Today, we're uncovering the very blueprint of duplication, and we've got some incredible, silky smooth 3D animations to help us visualize it. So let's set this process into motion.
To truly grasp our own biology, we really have to ask ourselves a fundamental question. How does a single-celled organism eventually become a trillion-cell organism? Or, on a more relatable level, how does a paper cut actually heal? Well, the secret lies entirely in how our cells perfectly replicate and then physically split our genetic material. Think about it. If the resulting cells weren't completely identical, our bodies just wouldn't function. It'd be chaos.
So here's our roadmap for today. We're going to cover the purpose of cell division, check out the anatomy of a chromosome, run through the stages of mitosis, hit the final split called cytokinesis, and wrap up by comparing all this to binary fission in bacteria. Let's get to it.
Part 1. The purpose of cell division, essentially the building blocks of life.
So why do we even need this? The crucial point here is that mitosis is the engine running inside your body at this very second. For multicellular organisms like us, it handles a few highly specialized, absolutely vital jobs. It drives our physical growth, it manages the constant replacement of old and damaged cells, which, by the way, is exactly why your skin doesn't just wear away over time. It repairs tissues when you get hurt. And for certain organisms, it's literally how they pull off asexual reproduction. The whole system is dedicated to making perfectly identical cells so everything keeps functioning flawlessly.
Moving on to part 2, anatomy of a chromosome. Let's establish our main players.
Okay, to understand replication, we really need to look at the structure of our genetic material. Check out this crisp 3D render against that dark slate gray background. This whole structure is what we call a chromosome. See those long vertical strands in blue and pink? Those are the sister chromatids. It takes two of these sister chromatids, one inherited from each parent, to make up this classic single chromosome shape.
And this brilliantly illustrates the actual replication. Before a cell can divide, those chromatids have to duplicate. Notice how the original single chromatids on the left replicate to form the homologous chromosomes on the right. And you'll see they're joined tightly together right in the center by that glowing green dot. That crucial connection point. We call that the centromere.
All right, section 3, the stages of mitosis, commonly known as the PMAT sequence.
Here is the precise step-by-step choreography of cellular division. It all kicks off with interphase, right before mitosis officially begins. From there, we flow smoothly through the 4 highly active stages of PMAT: prophase, metaphase, anaphase, and telophase. Let's start with interphase. Historically, textbooks called this a resting phase, which honestly is a terrible name. Behind the scenes, this cell is working overtime. It's growing larger, it's replicating essential organelles, and most importantly, it's duplicating its DNA into those identical sister chromatids we just looked at. It's basically gathering all the resources it needs for the main event.
Then, boom, the action really begins in prophase. You can think of this as the preparation stage. Watch how the protective nucleus entirely disappears. Meanwhile, those newly duplicated chromosomes tightly condense. They transform from a loose tangle into these thick, distinct structures that are clearly visible in the cytoplasm. At the same time, specialized spindle fibers start forming around them.
Next up is metaphase, and the M here easily stands for middle. The nuclear membrane has completely broken apart now. And look at this. Those glowing spindle fibers reach out and physically attach themselves right to the centromeres. They gently pull and push the chromosomes until they line up perfectly, straight down the middle of the cell along the equator.
Building up maximum tension, anaphase hits. The A stands for apart and away. The centromere officially divides, and those attached spindle fibers rapidly shorten. They forcefully pull the single identical chromatids apart and away from each other, dragging them smoothly to completely opposite ends or poles of the cell.
The dramatic movement finally wraps up in telophase. The T stands for 2. Now that our chromatids are safely at opposite ends, 2 brand new nuclear membranes form, wrapping protectively around each newly separated set of chromosomes. The chromosomes start to uncondense, losing that thick shape, and those spindle fibers that did all the heavy lifting, they simply break down and dissolve.
Section 4, cytokinesis, the final split.
Look at how beautifully this unbroken visual sequence flows from left to right, from the preparation of interphase straight through the silky motion physics of the PMAT sequence, leading us right to cytokinesis. Here, the cell membrane itself smoothly pinches right in the middle. It completely splits the cytoplasm, officially creating two totally separate, identical daughter cells. And remember, for complex organisms like us, each newly formed human daughter cell contains exactly 46 chromosomes.
Finally, Section 5, binary fission in bacteria, or as I like to call it, the simpler split.
This side-by-side really highlights the contrast. While mitosis serves diverse roles like tissue repair inside complex eukaryotes using a true nucleus, bacteria take a way more streamlined approach called binary fission. See, bacteria don't even have a nucleus. Their single, circular chromosome just sits in a specialized area called a nucleoid, and their main goal isn't complex tissue repair. It's just rapid, sheer reproduction to massively increase their population.
The process is wildly efficient. In binary fission, DNA replication enzymes start at a specific spot called the origin of replication. As a copy is created, these two origins physically pull towards opposite ends, and the whole bacterial cell elongates. Then, a crosswall forms right down the middle, and it simply pinches straight into two fully formed bacteria. No glowing spindle fibers, no dissolving nuclear membranes, just a fast, hyper-efficient split.
To wrap up this explainer, whether we're looking at the gorgeous structured PMAT choreography happening in our own bodies or the rapid-fire binary fission in bacteria, the ultimate goal is exactly the same: flawless genetic duplication. Mitosis uses PMAT and cytokinesis to ensure your new cells are perfect carbon copies, while binary fission ensures bacteria can multiply at an astonishing rate.
Think about that incredible blueprint operating silently inside you every single second of every day. Without you even noticing, your cells are perfectly copying their DNA, lining up, pulling apart, and smoothly pinching into new life, keeping you alive and growing. So as you go about the rest of your day, I'll leave you with this. What unseen masterpieces are the cells in your body building right now?